molecule optimization followed by a preclinical evaluation, clinical research, then only FDA will review our drug whether it can go to market or not. It has to go through these many processes to come to the market, and in that very initial stages lead molecule optimization. In that lead molecule optimization, like till now we have used traditional drug discovery method, which is like very expensive and how to say, like large number of molecules we have to synthesize. So cost effective also, like it takes more cost. So to synthesize 10,000 molecules or 1,000 molecules, it takes more number of time and cost. So to reduce that, we are using computer aided drug design, which stimulates our body in computer or whatever we need, like ADMET parameters or whatever parameters we need, what are the qualities we need in drug, we can check through computer aided drug design. Okay, so in the lead molecule optimization, there are two parameters, which is traditional drug discovery and computer aided drug discovery. And that computer aided drug discovery is a like systemic process which reduces the time and cost of a drug discovery process. Okay, then what is mean by CAD? How it is working? The main objective of drug discovery is not simply to identify a molecule that binds with the protein. Okay. How the drug is acting in our body. We are consuming paracetamol, for example. That paracetamol is entering into our body. We are swallowing the paracetamol tablet. It goes to the systemic circulation. Then it reaches the site of action. Right. In the site of action, it goes and binds with the cusp inhibitor or the respective target. So when our drug molecule is going and binding with the target protein, then it produces the activity. That is what is happening in our body. Okay, for example, you are having paracetamol. Okay, the paracetamol is having some side effect. Okay, but it is having an activity. If you take any cancer drugs, it is having so much of side effects, right? So the drug is producing activity, but the drug is having side effect as well. So how to overcome that problem? While designing a drug, it's while designing process itself, if we check ADMET parameters, toxicity profiling, everything, if we do carefully, we can overcome of those problems which is coming after post marketing surveillances. Right, because discovering a drug, it takes almost more than $1 billion of amount. We are spending more than $1 billion of amount to identify one drug, and that particular drug, after post-marketing surveillance, if it is getting rejected by FDA, we are facing so many problems. That 1 billion or 2 billion, whatever we spent, is waste. Right. So to overcome all these problems, when we design the drug, we have to be very careful. What are the parameters? What are the qualities should be there for a drug? In traditional method, we won't be able to identify that. After post marketing only, we will come to know, or during preclinical or clinical trials only, we will come to know the problems. What are the problems are there in the synthesized molecules? Why? Because we are just synthesizing those molecules and we are just treating with the animals. In animals also, we will complete the studies within 6 months or 3 months. Then we are giving it, whenever we are giving it to the humans, then only we will come to know these are the problems are there. So if you spend $500 million also, that is waste. To overcome those problems, computer aided drug design gives a proper solution for all of these problems. Okay.